So with that introduction, what else I will do now in the last sort of next 5 or 10 minutes is just give you a bit of background of the motivation of these designs. So that's. While I do that, I should say that we were pleasantly surprised that this workshop was oversubscribed and there were people on the waiting list, so that was a nice outcome for us. So I'll spend 10 minutes on the motivation for multi-arm, multi-stage trials. Two of the major challenges that face us in many areas, not just in the area that I come from of oncology, but in many areas, is to improve the success rate of our new therapies and speeding up the evaluation of our new therapies. I'm just looking around the room, I can see that there are a number of you who actually do studies, and those of us who in the room who do clinical trials know about these problems, that many of them are not successful in showing that new is better than old, and often they take a very long time to do. So if we just take oncology as a model for these issues, we've seen over the last 30 years that considerable progress has been made in improving cancer mortality rates. Many of those bits of progress have been through clinical trials, testing new therapies in clinical trials and showing that they're better than what we currently use, and then using those therapies in practice. Cancer mortality rates in many developed countries around the world are going down, particularly in breast cancer, but now in many other cancer types. It's a direct consequence of the new therapies that have been tested in trials. However, progress has been slow. It's taken us about 30, 40 years to achieve this. And more recently, you will have heard of targeted therapies or molecularly targeted therapies. Many of the phase 3 trials of those therapies have been, although apparently initially promising in phase 1 and phase 2 trials, the phase 3 trials have been very disappointing. Many have shown no effect, and those that have shown some effect, often that's been very small, that effect, across a range of tumour types. What else has changed over the last 15 years at least? I've been involved in this area for about 25 years now, is that we have many new treatments to evaluate now. 10, 15 years ago we didn't. So probably in those days it didn't matter that we took a long time to answer a question and answer only one question, and although we were disappointed, it may have not mattered so much that it was negative, because we didn't have too many. The opportunity cost was not great because we didn't have too many other opportunities. Now the situation is quite different. We have many new treatments to evaluate, and most of you are statisticians in the room, and you will hopefully acknowledge that as a large proportion of our previous therapies have been ineffective, it's likely that a large proportion of our current therapies that we're testing, the new therapies, are likely to be ineffective. A small number are likely to be effective, and our job is to weed out those small number that are effective from the large number that are likely to be ineffective. Not only are there many new treatments to evaluate, there are many aspects of treatments to test. Dose of treatment, duration of treatment, combinations of treatments with other treatments. So you can see the numbers of questions that we can now pose sensibly and appropriately is much greater than it's ever been. And that's a very, very good thing, of course. That's a direct consequence of our increase in of the increase in biological understanding of many of our major diseases. Alongside that, because of the changes in biological understanding, we've got diminishing populations. The increasing recognition, particularly in oncology, but in other diseases as well, of heterogeneity of disease. I mean, if you think away the way oncology defines its diseases, it's very simple. It's by the organ at which the disease occurs and the cell type. They're the two fundamental ways in which we describe disease. That's a very simplistic way of defining disease and determining approaches to both diagnosis of that disease, but also treatment of that disease. And as I said earlier, we've also got targeted therapies which are coming online, which hopefully will be targeted to groups of patients who are most likely to benefit. Whether we'll achieve that or not, we'll see. So that's a context for this. The other context, which I've sort of mentioned, but I've just put some figures on this slide for you here, to show that new is usually not better than the current. Both in academia and in industry, we've found that the success rate of our phase 3 trials is of the order of 30 to 40%. So of the order of 30 to 40% of our phase 3 trials that we do in academia and industry seem to show that new is better than the standard. That's quite a galling result for many of us who do trials, because we spend all of our lives doing trials, and any single clinician will probably lead maybe 3 trials in their lifetime. He's got a 60-70% chance of each of those trials being negative. Probably most of them, all 3 of them, quite likely to be negative. We as statisticians, those of us who are statisticians, we work on many more trials, so we have a higher chance of success. But even looking around the room, I know that a number of you are statisticians, and I know you'll have had your fair share, even more than your fair share, of trials which have not shown you is better than standard. And we spend 5, 6, 7, 8, 9, 10 years on each of these trials. I certainly want to find ways that I can maximise my chance of working on trials that are going to improve outcomes. One solution, and certainly not the only one, and one we're going to talk about today, are innovative adaptive designs. And of course there are many types of adaptive trials, and there's a huge methodological literature in this area. Actually, when you look at it, relatively few have been used. They've been largely used by industry, not by academia, not by us in the audience. Most of us in the audience are academics, although I should point out that we have a number of industry partners here as well today. But academia has really not responded to them very well. And as time is developing, though, academia is turning its attention to these adaptive trials, but mainly in the early phase setting, in the phase 1, phase 2 setting. We are talking largely today about the phase 2, phase 3 setting. 2 that academia have both developed and used, I highlight there, but as I say, there are others now being developed, and we may pick some of those up in discussion. The multi-arm, multi-stage trials, which is the focus of today's workshop, and the I-SPY 2 trials, which are sort of phase 1, phase 2 trials developed by the MD Anderson and others. And just to give you a definition, I thought this definition of an adaptive trial was a very good one, to use the accumulating data to decide on how to modify aspects of the study without undermining the validity or integrity of the trial. That's a very useful definition of an adaptive trial. The multi-arm, multi-stage trial, and I'll just introduce this briefly so as we're all start in the same place. A number of you will have seen these before, so I won't spend too long. The idea is set out here, firstly on this left panel where I've put the traditional approach. What I've shown you there are 4 new treatments, T1 through to T4, each being tested in the phase 2 settings, starting at different times, and then those that are successful, however you define success, in the phase 2 setting, are then continued and ran to a phase 3 trial and compared against the control arm in a phase 3 setting. And T2 happens to fall out at the phase 2 stage there, and the others carry on. The multi-arm, multi-stage idea is very simple. It's very, very simple, that you just do all that concurrently. You stack up all your new treatments T1 through to T4 and your control arm from the start. You do your phase 2 and your phase 3 components all in the same trial. You randomize from the start, and you include all patients for all the outcome measures that you use at all stages of that trial. So it's a very simple idea. It's not hard to see that if you could pull off the right-hand panel, multi-arm, multi-stage, rather than do the traditional approach, you'll be in a better place. You'll do things faster, you'll do things in a more efficient setting, you'll find results contemporaneously, which is an important point, and you have more chance of success. You know, you don't need to do fancy statistics to show all those points emerging. And there are some of the points I'm there, and one of the interesting points about these sorts of trials is the idea that there's fewer delays and only one application you have to make. All of us who do trials know that it takes forever to put these applications and protocols together, and then to go through all the processes that you have to get them approved. If you can do it all in one, it's much more efficient and quicker than doing many different applications. One of the things we will talk about is the increased flexibility that this gives you as well. It doesn't also mean that you put too much emphasis on one new treatment. And that's an interesting point that we might bring up in the discussions. I don't think any of the speakers are going to make this, is that most two-arm trials go on to completion. They don't have any, rarely do many of them have futility or lack of benefit or lack of activity analyses. And that's because once you've set out on a road to test a new treatment, you want to really make it sure that you give it its best chance of showing an effect. And most people don't want to ditch out at an earlier stage, given that they've got to where they've got to, spent all that time and energy to get to that stage. We might discuss that. And also, we're now thinking, we will present some data on adding arms as the trial continues. So I'll stop there.